Autophagy is a highly regulated cellular process governed by a network of genes and signaling pathways that ensure the body's ability to maintain internal balance, especially under stress. Among these genetic regulators, certain core autophagy-related genes, such as ATG5, BECLIN1, BECN1, and others in the ATG family, play indispensable roles in orchestrating the formation and function of autophagosomes, the cellular structures that engulf and degrade damaged organelles and macromolecules. When these genes are disrupted, either through inherited mutations or epigenetic changes, the entire autophagic system can become impaired. This genetic breakdown has now been linked not only to neurodegeneration and cancer, but increasingly to obesity and metabolic dysfunction. ATG5, for instance, is crucial for the elongation and closure of the autophagosome membrane. Studies in mice have shown that deleting ATG5 specifically in fat tissue results in abnormal fat accumulation, enlarged adipocytes, inflammation, and insulin resistance, even without a high-calorie diet. Similarly, BECLIN1 is involved in the initiation of autophagosome formation and in regulating crosstalk between autophagy and apoptosis. Reduced expression or activity of BECLIN1 has been observed in individuals with obesity and is associated with increased fat mass and hepatic steatosis. These findings indicate that genetic disruptions in the autophagy machinery can independently drive the development of obesity, not merely as a consequence of excess calories, but as a fundamental defect in cellular housekeeping. What makes this more concerning is that the downstream effects of impaired autophagy are systemic. Without efficient autophagic activity, cells accumulate damaged mitochondria and toxic lipid intermediates, which trigger oxidative stress and chronic inflammation. These stress signals then interfere with insulin signaling pathways, leading to insulin resistance, one of the key drivers of obesity-related diseases such as type 2 diabetes and fatty liver disease. In fat tissue, impaired autophagy also prevents the proper turnover and differentiation of adipocytes, leading to dysfunctional fat expansion and metabolic inflexibility. Some human studies have now begun to link specific polymorphisms in autophagy genes to obesity risk. For example, certain variants in the ATG5 gene have been associated with higher body mass index, BMI, altered lipid metabolism, and elevated markers of insulin resistance. These associations remain even after adjusting for dietary habits and physical activity, suggesting a direct genetic influence on metabolic pathways. Moreover, epigenetic factors, like DNA methylation patterns affecting autophagy gene expression, may further modulate an individual's susceptibility to metabolic diseases, especially when combined with environmental stressors such as poor diet or sedentary lifestyle. The positive takeaway here is that even when genetics predispose someone to impaired autophagy and metabolic dysfunction, the system remains plastic and responsive to lifestyle interventions. Regular exercise, fasting protocols, and plant-based bioactives, such as polyphenol or omega-3 fatty acids, have all been shown to upregulate autophagy genes and restore function in individuals at genetic risk. In some models, enhancing autophagy pharmacologically via mTOR inhibitors or AMPK activators has been able to reverse fat accumulation and restore metabolic health, even in organisms with genetic mutations. Understanding the genetic regulation of autophagy provides more than just insight into the roots of obesity. It offers a framework for precision medicine. In the future, individuals with known polymorphisms in ATG genes or reduced autophagy capacity might be identified early and guided towards specific interventions that activate or support autophagic function before obesity and its complications take hold. This genetic lens could also help explain why some people struggle with weight despite healthy lifestyles, because the issue may lie not in willpower, but in underlying cellular maintenance defects. That said, the warning is clear. Genetic impairment of autophagy is not benign. Left uncorrected, these disruptions can trigger a cascade of cellular dysfunction, contributing to fat accumulation, hormone imbalance, and chronic disease. But with the right tools, nutritional strategies, physical activity, targeted compounds, and future therapies, it is possible to bypass genetic obstacles and restore a significant degree of metabolic function. In conclusion, mutations and regulatory defects in autophagy-related genes like ATG5 and BECLIN1 are increasingly recognized as root contributors to obesity and metabolic disease. They weaken the body's ability to maintain internal order, leading to fat storage and metabolic chaos even in the absence of overt lifestyle errors. However, this risk is not destiny. With targeted, informed interventions, it may be possible to restore balance and resilience, even in the face of genetic predisposition, by awakening and supporting the body's own repair systems.